this video, we will continue our discussion about the relationship between KRAS mutations and FAS receptors on the pancreatic cancer cells. In our previous videos, we have discussed that when the KRAS receptors are when the KRAS mutations are being reduced or eliminated, then it will lead to the increase in the FAS receptors on the pancreatic cancer cells, and then it can interact with the immune cells that can trigger apoptosis on the pancreatic cancer cells. And we have seen decrease in the pancreatic tumors within the pancreatic cancer cells where KRAS mutations are being turned off. So in order to validate these things further, the researchers have used another approach, that is to suppress these FAS receptors and see whether the pancreatic cancer cells again starts to grow in the pancreatic cancer cells where KRAS mutations are off, or there is some another factor that is also playing a role. In order to do so, they have used short hairpin RNAs that can target these FAS receptors, and they have tested different short range RNAs that are listed here. And in the panel A, what they have presented here is that first they have checked whether the short range hairpin RNAs worked against these FAS receptors, and simultaneously they have also checked which short range hairpin RNAs work maximum or will be able to reduce these FAS receptors to a greater extent. So this is the control where the KRAS mutations are on. So the KRAS mutations will work towards reducing the FAS receptors. At the same time, the short range hairpin RNAs will also accelerate it or support that functions. And when the KRAS mutations are turned off, we can see that in the control there is an increase in the FAS receptors, whereas when different short range hairpin RNAs are used, they can see the decrease in the FAS receptors. And they have seen that for this SHFAS 28 short hairpin RNAs number 28 will be able to provide the least number of short range hairpin RNAs compared to the control. And same things they have observed when the KRAS mutations are turned off after 5 days. And this is the Western blot, where also we can see that for the short hairpin RNAs number 28 is turned on, then there is almost negligible amount of FAS that can be observed. And thereafter, after validating that their short hairpin number 28 works well, they decided to use this short hairpin number 28 in the mice models. So this is the experimental process that they have used. That is, for the control, they keep the KRAS mutations are turned on and observe the MRI at this 10-day and 20-day until its survival. And similarly, they did it for the mice where the KRAS mutations are on till day 10. After that, these short hairpin RNAs are being triggered or activated using doxycycline treatment. Where the KRAS mutations are the actual experimental is that they have introduced these tumors that has this short range SCR and at the same time short range FAS number 28, and they keep the KRAS mutations on with the doxycycline treatment till day 10. After that, they stop the doxycycline treatment and check the effect on the mouse model of which has this activated short range hairpin against FAS receptors. And these are the MRI results. So first row they represent the MRI results at day 10, and they can see that in the control with this short hairpin RNAs against FAS receptors are on, they can see some of the tumor cells. And when the short hairpin RNAs are on and off, then they can see the decrease in the tumor cells. And when this short hairpin RNase number 28 is turned on, they can further see slightly decrease but almost similar. And when it is turned on and off during the period of these 10 days, they can see the further decrease in the short hairpin FAS receptors. And they can further see the decrease in the pancreatic tumors here. So this decrease is compared to the control case. And when they did it for 20 days, they can see that in the normal control case they can see increase in the pancreatic tumor, whereas for this when this short hairpin against FAS receptors has been turned on and off, they can see the maximum amount of reduction in the tumor cells compared to the case where when this short hairpin RNAs against FAS receptors are turned on the day 28, or not on the day 28. Short hairpin RNAs number 28 is turned on against the FAS receptors. This is the unusual result, as they can see that increase. They can see, yeah, when it is turned on, they can see that in the area there is almost no tumors. Tumors are actually represented by this dark white color. So here they can see no dark white color, whereas in this they can see the high white color, and here almost negligible. And here in this case also they can see when the KRAS mutations are on and off, they can see some amount of tumor cells. When they quantify these things using percentage change in the tumor volume, they can see that compared to the parental case, when the KRAS mutations are on and off, they can see the decrease in the amount of tumors compared to the case when this SRH is on. One thing I need to clarify. So it's not the white dot that is represent the tumor; it's the whole periphery that represent the tumors. So when this silencing FAS receptors is turned on, then the FAS receptors on the pancreatic cancer cells goes down, and they will increase the amount of tumor cells. And similar with the case for 20 days, they can see the high increase in the tumor cells. So now you might have understood this MRI figure. If you have not understood this MRI figure, you can comment it down. I can explain it in words. So let's go back to this panel E, where when this silencing short hairpin RNAs are on, they can see that increase in the increase in the volume of the tumors compared to the case where it is on and off, where the increase in the number of tumors is almost negligible, or it is comparable to this parental on and off. And when it is completely remain on, short hairpin RNAs number 28 against FAS receptors completely remain on, then they can see the high increase in the amount of tumors. And also when it is remain on and off, then they can slightly decrease, because effect the amount of time that which these short hairpin RNAs spend spent in activated conditions is less compared to the case when it is completely activated for the whole period of time. So this results also indicates that reduction in these FAS receptors will reduce the immune efficiency, and it will cause the increase in the tumor volume. Thereafter they check the survival rate of the mice, and with the KRAS mutations are on, they can see the decrease or lowest survival rate, and it is comparable to the case where this SCR, or this comparable to the case where the short hairpin RNAs against FAS receptors is remain on. And however, when it is remain on and off, that is short hairpin RNAs remain on and off in the whole duration, they can see slightly increase in the survival rate of the mice. And this dash line indicates the short hairpin RNAs number 28 remains on and off. They can see the increase in the survival life of the mice. Whereas when it is completely remain on, then there is a decrease in the survival lives. And when these shRNAs remain, when this again different short hairpins RNAs against FAS receptors remain on and off, they can further see the decrease in the life of the mice model. So this is this difference. They have not mentioned whether it is significant, statistically significant or not, because ideally this pink line should be below the green line if it has to be in line with this other previous data. However, when this shRNAs remains on and off regularly, then, and yeah, here it is, they have mentioned that the pink and green line, we can see that whether it's significant or not. So this pink and green line, they have not compared about this shRNA on and shRNA 28 on. However, when they, so let's just continue our discussion. So when they did parental on and off, then its lifetime becomes slightly more, or its survival rates increased to a greater extent when the KRAS mutations are on and off, or off after a certain time. And similar case they have observed for this shRNAs remains on and off. So these two are non-significant. So we can see that survival rates does not goes down. So this indicates that the FAS receptors plays an important role towards immune activations. When they quantify this tumor percentage in terms of the weight, they can see that when the parentals are on and off after a certain time, they can see a decrease in the tumor. Similarly, they have seen a decrease in the tumors when shRNAs are on and off, that is shown by here, off after a certain period of time. And when this shRNAs also remains on, then they can see the increase in the tumor volume, and when it is off after a certain period of time, they can see the decrease in the tumor volume. This indicates that FAS receptors plays an important role towards immune activations, and this is the immunoblotting that indicates the CD4 and CD8 plus cells along with TAPI. We can see that when the KRAS are on, there are less immune cells, and when this KRAS is off in the control, they can see the high immune cells. And when this SHSCR or short hairpins against FAS receptors, short hairpin RNA against FAS receptors works, they can see the decrease in the or almost similar number of immune cells compared to the parental case. However, when this short hairpin RNAs against the FAS receptors number 28 is on, then they can see the significantly decrease in the number of CD4 and CD8 plus cells. And these things they have quantified in panel I, where we can see that increase in the number of CD4 plus cells when the parentals are KRAS mutations are off after day 5, and similarly they have seen the increase in the number of CD4 plus cells when these shRNAs are turned off after day 5. However, the CD4 plus cells are non-significant when these FAS receptors turned on and turned off after day 5. It's non-significant. And for similar result they observed for CD8 plus cells, that when the after day of 5 day it is remain off, then they can see that increase in the number of CD8 plus cells for this KRAS mutations when this turned off after day 5, and similar for this short hairpin RNAs against these FAS receptors has been turned off after day 5. However, for these FAS receptors, they have not seen any significant difference when it is turned off after day 5. So these results are not in line with the what are the other results that are presented here, as they should see the increase in the number of immune cells when these FAS receptors are been turned, FAS receptors against 28 has been turned off. And so we will discuss these things in our next video. However, the difference is non-significant, but we can see that, yeah, I would like to clarify these things, that there is a increased trend, we can see that. However, this silencing FAS receptors is again silencing as short hairpin RNA's FAS receptor 28 is so aggressive that even it is turned off after 5 days, it will take some time for the immune cells to again starts to develop and act against the FAS receptors. And that's the reason when this short hairpin RNAs number 28 against FAS receptors turned off, the effect will take a longer durations to show that increase in the number of FAS receptors and the immune cells are working against it. So this could be one of the reasons why after 5 days, even after 5 days, it is still, the immune cells are still not have picked up. So I hope you got a good and basic understanding or good understanding about these FAS receptors and the KRAS mutations on the pancreatic cancer cells. If you need to understand more about this in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.